All right. Well, thanks to the organization for inviting us. I'm very happy to be here and to talk to you guys a little bit about what we do at Bristol Myers Squibb. I think some of this is going to be somewhat redundant as what we have talked about this morning. So I don't have to go into details on the different details of the different assays that much. But really, I guess what I wanted to do is to see how these kinds of assays that we have described, how in particular we apply those to the drug development pipeline at Bristol Myers Squibb. All right, so the usual, pretty much all biologics have the potential to cause an immune response, and almost all do. And the hallmark of immunogenicity is obviously the anti-drug antibodies. I do like to show. Let me see. Is this the? No, this is not it. This is the point. Yeah, I do like to show that obviously early on when we started making antibody drugs, we optimistically went into the clinic with fully murine antibodies and quickly encountered the problem of immunogenicity as being a somewhat substantial hurdle for biological drugs. Either doing some engineering or humanizing, or even when we arrived at the fully human antibodies, does not seem to have fully reduced this problem. So even though we as a company, most of our antibodies are actually fully human antibodies, we still have to be careful about immunogenicity. And that can vary case by case. So impact, as we talked about, impact for immunogenicity vary anywhere from non-detectable, as talked about this morning. For Bristol Myers Squibb, we have a bunch of immuno-oncology products, the anti-drug antibodies to particularly the anti-PD-1 antibody, especially in combination, seems to be nicely increased, but we don't see an impact on efficacy for those molecules. So that's, I guess, the best case scenario for when you have anti-drug antibodies. But, as also shown, particularly this morning by Amy, you can have some serious adverse events. So you have to pay careful attention. Neutralization, I think, is particularly, if we're talking about fully human antibodies, the CDRs are really the only remaining portion that is non-self, or we like to consider non-self. Therefore, if you get anti-drug antibodies, they're likely to bind there. If they bind there, since that's the portion of your molecule that is involved in target binding, you're quickly faced with neutralization. And that's why we have these NAB and antibody assays in the clinic. Changes in PK, that's the other big thing, and that we're obviously paying close attention to. You can either get an increase in your PK for very small molecules, but I think in most cases what you see is you get, once those anti-drug antibodies start appearing, your PK starts dropping off. You quickly clear your drug, therefore you lose efficacy, and that's why we pay close attention to once we observe anti-drug antibodies in the clinic, we try to correlate, does that have any effect on the PK and efficacy. And lastly, as mentioned, why this is important to look at. It can have loss of function is obviously important for your patients, but also for us as companies, because cancer programs cost a lot of money and it makes nobody happy. All right, so just to lay out a little bit the complexity that we're dealing with, just as in the last talk, we kind of control the protein factors. We know what sequence we have in our drug. We know what the protein is. We know we can characterize the chemical modifications, glycosylation, stability, et cetera, et cetera. Some of the other factors, yes, we can control the drug factors. What container are we going to put it in? Storage. The patient factors are kind of dictated by what disease we're choosing to treat, or what the indication is. But that adds a lot of unknowns. And then obviously there's the treatment factors. What dose are we going to give? How frequently are we going to give it? route of administration and whether we combine our drug with other drugs. So if all those factors go into determining whether or not we see immunogenicity in the clinic, we kind of have to balance that with how, what is the potential benefit of this drug for patients. Obviously, some very life-threatening diseases have a much higher tolerance for immunogenicity than something that has already have competing products on the market that might have lower anti-drug antibodies. So that is kind of a balance that we like to strike in discovery, and determines on how much work are we going to put into immunogenicity, and how likely is it that the program will cancer? What is the perceived risk of immunogenicity? All right, so the strategy that we have that we usually apply within BMS, and this is really just for the preclinical early drug development work. So as mentioned, really there's no good immunogenicity, so it's pretty easy. The more we can minimize, the better. So any risk factor that we see, if we can eliminate it, awesome. You don't really have to balance with anything. So preclinically, that mostly means for us that we're looking at sequence-based liabilities. I think for us as an organization to understand immunogenicity in the clinic is very important, and that feeds back into the work that we do. So we want to have a good understanding of what happens later on with our products once they go into the clinic. And especially since we are a big organization, that is not always a obvious feedback loop. Since in early discovery, we take not just immunogenicity liabilities into account, but a whole host of other liabilities, chemical liabilities, manufacturability, stability, et cetera. So it kind of goes into that package that goes into consideration of whether or not we move a biological drug forward. So as mentioned, it's really a case-by-case scenario that we strike for each new product that we make. What is the immunogenicity risk versus what are the perceived benefits for our patients? And then lastly, we definitely like to use multiple methods to assess immunogenicity risk, mostly to avoid false positives, and multiple methods giving us indications in the same direction. So this gives us a lot more confidence in the assessment that we do. So kind of a variation on previous slides. This is really just the assays that we deploy in discovery. Here's the classic MHC class 2 antibody development. And really the tools that we use, so in order to see what the potential or what the sequence potential of our drug is to bind HLA, we have a in silico tool that we use, and then we have a MAPS assay. Once it comes to T cell activation, we have two variations. We have as described DC T cell assay, but we still do a good amount of straightforward PBMC assay, meaning we're not pulsing DCs. We're just adding our drug or antigen straight to a PBMC culture. Then just to look at innate activation, we do have some cytokine release assays that we can deploy to see what kind of activation we get there. And then lastly, we've done some experimental work just to see on, as also talked about, obviously B cells possess a particular problem to get maintained in a cell culture. And yeah, so those 3D artificial lymph node systems are very interesting. And we dabbled a little bit in these fully human mouse systems that have a reconstituted human immune system. And we do see some isotype switching and antibody development in these mice. It remains kind of for us, a question on where would we deploy these kinds of models. They're labor intensive, they're very expensive, so we still have to find a good use case for these kinds of models. All right, so the BMS drug discovery pipeline, so to say. So I went back and looked a little bit, so this is a rough estimate on what we do programs-wise. So in 2017, we had about 22 biologic discovery programs. As mentioned, the vast majority of those programs are all fully human antibodies. We have some engineered endogenous proteins. Obviously, those are, from an immunogenicity perspective, a little bit more challenging. And when it comes to disease areas, like most of our antibodies are all immune oncology products. So you guys have seen the PD-1 and the CTLA-4, and then obviously there's a whole set of other T-cell and ABC and macrophage-related checkpoint inhibitors and activators that we are working on. We do a good amount of the opposite. If we make an agonist, we might as well make an antagonist, and then our immunology folks like to look at those same targets. And then we are working on fibrosis and cardiovascular. Per year, I think I looked at 2017. So we ran about 18 assay runs. For that year was about 50% PBMC assays, 50% of those dendritic cell T-cell assays. I think right now we're trending more and more to exclusively running those DC T-cell assays. We'd like to get involved early. That doesn't work always. So some programs come to us and they only have one candidate that still works, that has passed all the other hurdles. So then we'll look at that one. But if the ideal situation would be that we look at multiple candidates and that we can advise the development team on what we perceive as the lowest immunogenicity risk molecules. Mostly we run full proteins, and because we're early on, they're easy to get then protein, and that seems to be the fastest way to do it. And we like to, if we get 15 different antibodies, we like to rank them and then move forward the molecules that look least riskful. Obviously, there's a good amount of engineering going on these days in the antibody world, it either being the FC or affinity maturation or pH sensitives, et cetera. We have a large set of very happy protein engineers that do not really take immunogenicity risk as their highest priority. So, but in collaboration with us, we try to guide them through those challenges. So then really what our goal is for our group is to provide, when we move a new biologic drug into the tox or first in human, we like to have a comprehensive immunogenicity package available for those folks, which means really we provide in silico and in vitro data. MAPS assay or some of the other assays is really as needed. And then we put that in context with the other risk factors that we discussed. So that gives the development team somewhat of an idea of where we think these molecules stand. And then lastly, the folks that are developing the clinical assays, we like to talk to them and work with them, and the risk assessment that we do kind of guides how they're going to apply their assay strategy once they go into the clinic. So that means that decides, are we going to look at ADA assay results in real time, or do we save the samples until later? At what point do we start developing our NAP assay, et cetera, et cetera? All right, so this is really the timeline that we work with, and I just wanted to show this to illustrate how our system works. So in early discovery, obviously people are finding out their target, and they're starting to immunize those human mice, and we start looking at hybridomas, and we're doing hopefully sometimes some sequencing of those antibodies. Unfortunately, we can't really throw hybridoma material straight into an in vitro assay. That gives us there's too much contaminants in there. But so early on, once we start getting some sequencing data, we can start doing an assessment of the antibodies. Then once they start moving, we start scaling up those antibodies a little bit. They become a little bit more cleaner, and we can actually work with them. And we start getting in vitro data from those antibodies. Obviously, if we get a similar kind of signal from those two assessments, that gives us somewhat more confidence. But then at one point, we pick one candidate that needs to be scaled up and go into toxin in the clinic. So that's when we provide that whole package. And then once we get some first in human data, it'd be really nice for us to get that back. That sometimes works, sometimes not. I think I have a slide where we from the data that we did get back from those guys. So some notes on the in silico tools that we use. So we actually run a in-house version of the IEDB tools. So it's 27 DRB1 alleles. You get a relative ranking from your 15 MER, one overlapping 15 MERs. And what we do is we actually, because our framework for the antibodies is fully human, so we really just exclude the whole framework of the antibodies. So really we do an assessment of what the CDRs are. Obviously, this is a fast and cheap way to go through your sequences. So really, it works if you have an abundance of sequence. It makes it easy to prioritize and do a selection. If you don't have that much, if we don't have that many sequence or antibodies, we rarely exclude anything just based on that kind of data. Obviously, it can identify potential protein engineering sites if you don't have too many hotspots in your antibody. And as mentioned, we use it for prioritization and ranking of clones. On the con for the in silico, as mentioned by previous speakers, obviously false positives is somewhat of an issue. I thought that the MAPS data showed that yesterday very nicely. So that's a challenge, and it really it's what to do about it. I guess we need better in silico tools. So really, if you get a hotspot in a in silico assessment, whether or not you actually get that peptide, we don't know. Whether that epitope is tolerated. So that makes it kind of challenging. Let me go quickly over, because it's very similar to what we previously shown. This is the dendritic cell assay that we run in-house. We take, it says 36. We actually do use 40. And our donors are actually locally here from Boston. Luckily, we have a diverse student population here in Boston from which we get our blood. So it should be a fairly good representation of the world population. That said, we still do HLA typing for those donors. We also CFSE label our CD4s. We do not, we just add PBMCs to our DCs. I think we use a fairly similar DC maturation protocol. We pulse those and then they get added to autologous PBMCs. After 7 days in culture, we look at the proliferation rate. So I do like to show this slide, and this is just really what we did is, we've run a good amount of antibodies. I think these are mostly antibodies, maybe a few non-antibodies that have gone into the clinic. And what we've done is we've just looked at what is the proliferation rate that we see in the in vitro assay, and how does that stack up to the ADA rate that we see in the clinic. Obviously, this ADA rate is just what we take off the label from those drugs. We all know the limitations of those data sets and whatever assay those organizations decided to measure that with. Nevertheless, I'm cautiously optimistic about the correlation that we see for this assay. So that makes it worth it for us to at least run our discovery molecules through these kinds of assays. Next slide. All right, go. Okay, so some notes on these in vitro assays, and especially interesting since what we have previously talked about here. So blood and donors. While some people like to deplete their CD8, we have decided to not deplete CD8. We go with straight PBMCs in our DC assays, or we don't add just CD4s. And I guess it might be interesting at one point we should all sit down and compare results and see if we actually get different results that way. The same thing, the frequency of the receptive T cells, it has been described to be somewhere in the 1 to 1,000, 1 to 1 million for CD4s. So really the bottom line is the more cells you can put in there, the better. So I was very impressed with Teva putting 1.5 million CD4s on their assay. We do 200,000 cells, PBMCs per well, and we run that in 6 replicates. I guess another note would be we found early on for development isolating fresh PBMCs and being very careful in what your procedures and handling is of PBMCs and keeping close track of your viability and recovery has made a big impact on our assay performance. And then lastly, the to pre-screen or not to pre-screen before you go run an assay, I think is always an interesting question. I've seen some people would like to say, if my donor doesn't respond to a model antigen, say KLH or something, then it's probably a bad donor. We're not going to use it. I think we've tossed that question back and forward several times, but I think we've seen enough donors that would not respond to the model antigen, but then manage to mount a response to one of our products. So we're not pre-screening anymore. So these assays are interesting from a analytical perspective, as in you don't always have the same 40 donors because you can't always get the same people. So repeatability of these assays is challenging. We like to run low and high control antibodies just to keep track on what our assay performance is, and then kind of also if we get an unusual result to at least go back to people and say, like, look, the assay didn't really perform that well this particular one. So so we do like to keep track of that and do trend analysis on our 2 control antibodies. So this is probably, I think this is somewhere in the 25 assay runs for these controls, and as you can see, this is the difference between our high and low control, percentage of positive donors, and pretty reasonable variation for those assay runs. So that also allows us to look if the control just starts looking very much out of that range, then we might decide to run it again. And that does seem to be a challenge for us. The repeatability or between different batches or outliers, etc. So I think it's a particular problem for discovery programs, and that's mostly we're working in early discovery. So these programs that we're working on, the purification train has not been fully developed. So there should be a good amount of impurities in these runs. So that sometimes results in pretty impressive batch-to-batch variation for those molecules. And as mentioned, I think that we all know that there's plenty of impurities that come along in your purification that can activate your immune system, and by conveying a danger signal will still result in proliferation of your CD4s, resulting in a false positive. I have some data here that we recently generated. So here's our low control. It's is it described? No, it's the Avastin antibody from Genentech. We run it pretty much in every run. It's nicely controlled, nice and stable, nice low response. When we try to make this in-house with a quick and dirty process, so to say, I think it's just a protein A purification, you can see that you can get a pretty good variation from those different batches. So that's a challenge that we're dealing with. I don't think we have it solved all the way. We're trying to identify, can we pre-screen these guys and see what kind of impurities are important. I don't think we can require all of our programs to do a 3-step purification train for all their discovery antibodies. So either that, or we can screen, pre-screen these batches with a cytokine release assay. But it remains somewhat challenging. So, as mentioned, let me switch to immuno-oncology products. Obviously the IO in checkpoint inhibitors have made great impacts in the clinic, and I think at this meeting we've seen some impressive data on them, being a new pillar in oncology treatment. From a immunogenicity perspective, and especially for us in early discovery, they pose somewhat of a challenge, as in they kind of mess with any of the cells that we like to throw in our plates, and then really we have to deal with the mode of action of those antibodies activating or inhibiting our T cells or DCs. So this is really just a recent picture from, I think, from Nature on what are all the co-stim and co-inhibitory molecules, and I think that most companies that are working on these kinds of drugs probably have the majority of these targets in the development. So really then we have to ask the question, how do we, if we're working on those antibodies, how do we want to assess the immunogenicity risk of these guys? As said, the mode of action kind of for the different antibodies works on every step of our in vitro assay, so that makes it kind of challenging and can result in either false positive or negatives. Interestingly, we have found that just because an antibody should be activating does not necessarily mean that in the assay format that we like to run that they are inhibiting these assays. I think because we are running such high concentrations, clustering effects or other mechanisms can either deplete your particular cells or activate cells that you didn't expect. So unexpected things happen, and it makes it tricky to actually figure out what is the, I like to call it intrinsic, but what's the sequence-based immunogenicity risk of those antibodies. So what we like to do for those kinds of molecules, we do like to run a KLH interference assay. It's kind of analogous to what the tox folks do with a TDAR assay, really, where you look if your drug either inhibits or enhances the antibody development in your tox animal. So we figured we could probably do something similar in vitro, and we're just gonna run KLH, and then we add our drug, and that will give us somewhat of an idea. And we don't have to do it with that many donors because it's pretty obvious. But it gives us an idea whether the mode of action of our drug messes with our assay format. And we can do that either for the PBMC assay or the DC assay format. So really what that looks like is, you get a good response for KLH, and you can, if we take these different kinds of IO antibodies, here we have an example of what it would look like if the mode of action is somewhat inhibiting, and it really knocks the crap out of it pretty well. And then actually, I would say more often than not, we see inhibition versus enhancement. We do have some examples where we see a pretty good enhancement. So once we have this data, we have several options. Like if we see inhibition in the PBMC assay, obviously we can go to a DC assay that will not expose your antibodies to T cells. If you also see a effect in your DC assay, you will have to consider something else. So that might either be maybe you should use another cell type as uh-oh. Is my time up? All right. So I'll speed it up a little bit. But that gives you then it gives you the option to say, I'm going to use another cell, I'm going to use overlapping peptides, or maybe I'll just use the MAPS assay. So MAPS assay we have talked about. So there's not much I have to say about it, but I think it's a very nice solution, and it provides you with a perspective of what your APCs are actually presenting. Besides that, I think that the MAPS assay, once we start collecting that data, this might give us a very nice opportunity to further enhance our in silico models, just because it will give us real-world data of what your antigen-presenting cells are going to present, and then in combination with the advances that we are making in machine learning, if we integrate all the data that we have on biologics processing and what gets presented, hopefully this should allow us to develop some better in silico models. All right, so I'll quickly run through the last thing here just to show the challenge of de-immunization. As mentioned for discovery, this is our discovery train. This actually gives us a very small window to do any work on. So really the reality is we do an assessment and then they're off to the clinic. That's challenging. So mostly we do ranking, and obviously we encourage people to make a lot of clones. That's a much faster way to find low risk clones. And then protein engineering obviously has the challenge that if you start de-immunizing, and one of our strategies comes up always is can we just germline the CDRs? That's interesting, but then obviously you're messing with your CDRs, and that gives you the risk of disrupting binding. And then for in silico tools to de-immunize, you really need a good amount of structure function data in order to use that. So my example here is, here we have an antibody. This is the in silico assessment. Here you can see this is the V heavy. We got pretty good hotspots in the CDR2 here. When we and then here this random spot here. So when we did some engineering on this guy and germlined these, we saw some interesting observations. So here you have the wild type score, and then we made some substitutions, some germlining in this CDR here, and that reduced the in vitro score of these molecules. Unfortunately, that also abolished binding, not unexpected. Another example is this other clone, this antibody 2 here. We made a substitution here in the framework, but I guess this was a false positive because it didn't really change any of the in vitro score. We only have one substitution here that's on the side of this CDR3 that seemed to reduce the score pretty well, and it retained binding. So that was kind of a lucky shot, and I think we don't really fully understand it yet. So now that we have a MAPS assay in place, this is the same antibody. I got the net MHC score here. This is our in silico tool in-house. And yes, we pretty good get the peptide back for this particular CDR2. So that gives us a pretty good target to where we could start working on protein engineering this molecule. So this is really this is my last slide here. And this is what we're currently working on: a proposed workflow for a second generation molecule where we would de-immunize these molecules. So really, you have an antibody that has some immunogenicity issues. You use the MAPS assay to identify what areas is an antigen. since we come from a in vitro display technology company, we do have technology in-house to do this, create a library from the fabs and find what amino acid substitution are actually functional, and therefore what could be used to decrease immunogenicity. It's very analogous to the mute map technology that Jeremy described. So once we do that, we can reassemble some variants that we think have decreased immunogenicity. Obviously, we would check binding again and confirm with the in vitro assay, and that would be the way to get to a de-immunized molecule. But as mentioned, obviously, that's a pretty long timeline that right now doesn't fit in our development cycle. So most likely, it would have to go into a second-gen molecule. All right. So I'd like to thank these people from the group that did all this work, and Bristol Myers Squibb is moving to Cambridge. We're actually moving around the corner from here, I think in 2 months. So with that, I'll leave it and I'll take some questions.
Great, thank you.
All right.
When you talked about immune-targeted drugs.
Yes.
If your immune-targeted drug is something like a T-cell engager, does some of the techniques you talked about, the KLH binding, does that apply there, or it does?
Well, that's a good way to see how much of a interference would we get in the assay, or it would kind of separate what effect comes from the mode of action of the drug and what comes from the sequence-based.
Okay.
And we'd like to separate those.
Okay.
Right. So if we have a T-cell engager and we see that there's a big enhancement in the KLH assay, that would drive it to say, well, then we're going to try to pulse it into a DC T-cell assay. If we then still see it, yeah, then it becomes challenging, and then we can say maybe we'll try another cell line, maybe we'll try overlapping peptides. And since our drugs are mostly fully human antibodies, I'm somewhat comfortable by just using overlapping peptides that cover the CDRs.
And another question related to that is, and then somebody else brought it up earlier, is, I mean, going to patients who have been either, and because you're probably going to many of these immuno-oncology drugs directly into patients,
Yeah.
who've been heavily pre-treated or have had other regimens.
Yes.
And obviously modulation of their immune systems. Have you looked at any correlation between the immunogenicity data from that versus what you see from your maps or other assays?
Yes. So there's two things to that. I think the correlation slide does have several of those immuno-oncology molecules in it. The other thing I would say is, I think the notion that oncology patients are severely immune compromised doesn't really pan out for a large group of the oncology patients these days, especially with the washout period. I mean, and you can see we can still get pretty good anti-drug antibodies from oncology patients. So I think that plays into it.
Hi, great
Hi.
talk. So as you know, there's multiple platforms to generate antibodies, biologics. With your experience, did you notice or have any correlation with certain platform, fresh display, humanized mice are better that generate less immunogenic, more immunogenic molecules? Did you ever look at it?
That's a very interesting question. Yeah, we have definitely talked about it. Our main platform is a HuMap from these mice. So I don't really have any direct data on what a artificial library in phage display would look like. I'm trying to think back of if I heard anybody else mention that. I don't think so. No.
An additional question. So you mentioned that you don't look at the framework.
Right.
Right. But sometimes the framework is humanized to germlines that are not used, non-productive, and sometimes
Yes.
Engineering teams introduce a mutation that for liabilities, deamination, and so forth.
Yeah.
So how in the framework? So how do you?
So the way that the silico tool is set up is that it filters based on the frameworks that we have in the mice. But yeah, there's some and then we do some kind of blast to see if there's any rare changes, and if those are, they will show up. Yeah, so any of those kinds of changes we will look at. And then that's for the in silico tool, but then we will put the whole molecule into a in vitro assay anyway.
One thing that's very interesting about the low and high response comparators that you use as control or benchmarks in your assay. And I guess with Avastin, it sort of feeds out from our own experience. One of the challenges of these assays is that everybody wants you maybe to run the same protein again and again. But even if you have, I mean, in your situation, you've been obviously generating those different Avastin variants internally and
yes.
differences. But even when we're working with clinical batches of drugs, time to time we also see those differences. So actually, inherently, you've got that challenge that what should you do or not? Well, that's why it's, for us, looking at those different molecules on a per assay basis is really the only way you can do that comparison. Because otherwise, who's to say that batch 1 is right versus batch 3?
Yeah.
It's very, very different. And yeah, even on clinical batches, we see these
Mm-hmm.
differences.
Yeah, it's challenging.
What do you use for the high response?
We use a Pfizer IL-21R antibody. But yeah, no, obviously, and it goes, I mean, that's a challenge for anybody that does regulated assays. Once you switch your controls from one batch to the next, you got to qualify that it runs about the same. Yeah, we have seen batches where we say like, well, maybe we won't use this one. Yeah, challenging.
Great. Well, thank you very much.
All right. Thank you.